Good day, this is Professor Will, CBMD PhD. Today's Sunday, January 12th, 2020, 3:02:36 a.m. Eastern Standard Time. Today's reading, Transduction host DNA transfer by bacteriophages. So let's get started here. Genetic transduction is a transfer from a donor to a recipient cell of non-viral genetic material in a viral coat. Transduction is one of the three ways by which genetic material can be moved from one bacterium to another. The others are transformation, in which naked DNA is actively or passively taken up by recipient cells, and conjugation, in which cell-to-cell contact is initiated via structures encoded principally by plasmids. DNA is either transferred through these structures or protected by them during transfer. Both transformation and conjugation are evolved systems. Naturally transformable bacteria such as Bacillus subtilis, Haemophilus influenza, Streptococcus pneumonia have sizable sets of genes concerned with the transformation process. Conjugational systems are essential to the plasmid way of life, allowing plasmids to move between hosts, not necessarily of the same species, more or less promiscuously. Transduction, on the other hand, may be well simply an accidental concomitant of the way in which bacteriophages multiply and package their DNA. Transduction was discovered by Zinder and Lutterberg in the early 1950s, at a time when the existence of the gene was not yet universally accepted. Conjugation had been discovered in Escherichia coli by Lutterberg and Tantum in 1946, and it was conjugation transfer of DNA between two differently marked strains of Salmonella typhimurium that Zinder and Lutterberg were expecting to demonstrate when they discovered transduction. Conjugation had been shown to require cell-cell contact because it failed to occur when donor and prospective recipient were grown in the same vessel, but separated by a filter that allowed macromolecules and medium, but not cells, to pass through. On the contrary, Zinder and Lutterberg were to find that a filter did not impede gene transfer between Salmonella strains. Because it was expected that conjugational gene transfer would allow the co-inheritance of any pair of donor genes, however far apart, the strains tested were all doubly marked, i.e. contained mutations in two different genes to avoid mistaking reference or contaminants for recombinants. Although many strain pairings were tried, transduction was discovered only when a particular pair of differently marked strains of S. typhimurium was tested. This single success was a consequence of two happy accidents. We know now in transduction only linked markers can be co-inherited. By good fortune, one of the strains used, LT22, was both marked with two closely linked genetic markers and was lysogenic for a bacteriophage P22 that is able to transduce. Phages produced by spontaneous lysis of LT22 crossed the filter infected the other sensitive strain and carried transducing DNA home. Transformation was excluded as a mechanism by showing that transducing activity was resistant to DNase treatment, and further experimentation showed a phage to be responsible for the gene transfer. A few years later, bacteriophage P1, a lysogenizing phage of E. coli, was also shown to medicate transduction. Because any single genetic marker can be transferred by P1 or P22, the process is called generalized transduction. Transduction by these two phages continued to be an important genetic tool. In 1956, Morris Lutterberg described another sort of transductional system. They found that the bacteriophage lambda was able to transduce the galactose GAL genes, which are located next to the site on the chromosome at which the lysogenized phase is integrated. Because they could find no other genes that lambda would transduce, they named the process specialized transduction. It was later shown that the biotin bio genes, which with the galactose genes flank the lambda integration site, can also be transduced. Specialized transduction in modern guise has since achieved new importance because genetic engineering techniques permit the artificial construction of specialized transducing particles containing any desired gene. Specialized and generalized transduction are processes that are quite different in detail, but which have in common the fact that a bacteriophage acts as a vector to transfer host DNA from donor to recipient. Specialized transduction by coliphage lambda follows naturally from its temperate lifestyle. Lambda particles contain linear phage DNA with cohesive ends that result from staggered cuts within COS sites made during phage DNA packaging. These cohesive or sticky ends permit the DNA to circularize after infection. After circularization, a choice is made between two possible outcomes of the infection. In the lytic cycle, the lambda circle replicates rapidly, initiating a process that will result in the lysis of the host and release of progeny phage particles about an hour later. An intermediate in lytic growth is a DNA concatemer consisting of several full-length genomes arranged head to tail. This is the packaging substrate, which is a phage enzyme cleaves at successive COS sites to create the cohesive ended genome length fragments of fixed sequence that are packaged into phage heads. In the lysogenic response, the repression of replication is established and a chain of events is initiated at lead via a site-specific recombinational event between ATTP on the phage and ATTP on the host DNA. ATTP is located between the GAL and biogenes, so the integration of the phage into the continuity of the bacterial chromosome. Once integrated, it is fastly replicated once per division cycle by the host replicative machinery. It remains quiescent except for the continual production of a repressor that prevents entry into the lytic cycle. If repression fails or is interfered with deliberately by induction, the phage is cut out of the chromosome to reverse the process that occurred at lysogenization. If all goes smoothly, excision will be accurate, and a normal phage is generated. Specialized transducing phage arrives when excision occurs inaccurately to form a circle about the correct length, but with the wrong DNA. Provided that the replication origin is retained, such a DNA molecule can go through the replication cycle, including a concatemer formation, COS cutting, and packaging. Phages that have acquired GAL, lambda D GAL, where D signifies defective, will have lost phage capsid genes and be unable to undergo further lytic growth without help. However, because the cell in which the particle originates probably also contains complete phage genomes, excised perhaps from a sister chromosome, the requisite enzymes and structural components will be available to allow the assembly and liberation of defective transducing particles. Lambda biophages, in contrast, contain replication and capsid genes, and so are not defective. They will, however, lack some of the genes needed to attain lysogeny. Aberrant excisions that lead to recoverable transducing particles are rare. Only 10 to the power of -5 minus 10 to the power of -6 phages are of this type. If a lysate, such as the one described, is prepared on a GAL plus donor and used transduce GAL minus, recipient GAL plus progeny will be found at low frequency. These progeny will frequently be deployed for the GAL genes as a result of inheriting GAL DNA from both phage and host. Although haploid progeny can arise by the replacement of GAL minus and GAL plus, with loss of the phage. If the ratio of phage to recipient is low, cells receiving lambda D GAL will not be co-infected with an intact phage. Deployed GAL plus progeny will either be lambda D GAL lysogens with the phage integrated at the attachment site, or more often co-integrates of phage and chromosome generated by a single homologous recombination event between GAL sequences. These co-integrated GAL plus progeny will be unable because the integration is readily reversible. Both types of lysogen will be immune to lambda infection, but will not produce any phage on induction because they lack coat protein genes. Stable haploid, GAL plus, lambda minus sensitive progeny can also arise by the resolution of the co-integrant with allele exchange and the subsequent loss of phage DNA. If the ratio of phage to recipient is high, cells receiving lambda D GAL are likely to also be infected with a normal phage. In this case, dilysogens can form with the defective and normal phage in tandem array. The GAL plus phenotype of such progeny will be unable because homologous recombination between the tandemly repeated genomes will lead to frequent phage loss. Dilysogens are inducible because the normal phage encodes all necessary proteins for phage maturation. The product of induction will be a mixed lysate that can contain equal numbers of transducing and infectious particles. These high transducing lysates are not only convenient for further gene transfer, but provided a pre-cloning pre-PCR method for obtaining purified GAL sequences. In E. coli strains from which ATTP is missing, lambda can lysogenize at low frequency at alternative sites. This property was used to generate transducing phages for a variety of markers around the chromosome, before it became possible to use restriction endonucleases to create genomic libraries in vitro. Any temperate bacteriophage that integrates into the chromosome of its host should be capable of specialized transduction. This supposition has not been extensively tested. In vitro packaging allows specialized transducing phages to be made for any gene in vitro, and specially constructed lambda vectors are widely used for making genomic libraries for cloning particular genes. A lambda cloning vector is a phage that has been modified so as to provide unique restriction endonucleases cutting sites at the designated cloning site. Many vectors have inessential lambda DNA deleted to provide more space for cloned DNA and have lost the DNA needed to lysogenize. However, lysogenizing vectors are available in which DNA fragments several genes in length can be cloned, allowing such genes to be stably maintained in a single copy as part of a phage at the phage attachment site. Special purpose transducing phages of this sort have been made that permit the controlled expression of foreign genes in E. coli, such as, for example, phage T7 RNA polymerase, used to amplify the expression of selected proteins engineered to be transcribed by the polymerase. Other phages allow the linking of gene promoters or translation initiation regions to reporter genes, so that the expression can be quantified. Because the lambda lysogens contain single copies of cloned genes, problems encountered when gene products are overexpressed, as they usually are from plasmid clones, are avoided. In most systems, transduction is a rare event, with perhaps 1 over 10 to the power 5 phage particles able to transduce a particular selected marker. Because transduction is infrequent, so a single recipient cell will receive more than one transduced DNA fragment. Therefore, co-transduction will occur only if the markers are close enough together to be included on the same piece of DNA. Because transduced DNA is linear, it can become incorporated into the recipient chromosome only via a double crossover event. Because homologous pairing must be achieved, the requisite crossovers seldom occur very close to the ends of the transduced fragment, and in practice, the closer two markers are, the more likely they are to be co-transduced. Good day, this is Professor Will, CBMD PhD. Today is Sunday, January 12, 2020, 4:36, 16 seconds a.m. Eastern Standard Time. And this concludes my reading on transduction. Hope you enjoy listening. Have a great day.